right, welcome back. We're pushing through to the party tonight. That's what we're doing, right? So we are really, really pleased to have two amazing women in genetics to talk about Dup15q and answer any questions about Dup15q. We have on the line, we'll have Dr. Carolyn Shannon, who is actually one of the co-founders of the Dup15q Alliance back in the day when it was ideas and when it was based in Geisinger. She was one of the first people that connected the first sets of families for that had Dup15q. Back in the day, it was isocentric 15 because we didn't quite know about interstitial yet. So she'll talk a little bit about how that came about and how it was found. And then we have our amazing geneticist from Children's Hospital of Colorado here, Dr. Jessica Duis. And we'll go a little bit deeper into the mechanics of Dup15q syndrome. So welcome, thank you.
Carolyn's going to start, so she's going to share her screen.
I am sharing right now. Okay. Hi, everybody. I haven't done this way before, so hopefully this works out. And I'm going to warn you in advance that if you hear purring or barking, my animals tend to bomb me every time I ever start talking to my computer. So first, I want to kind of give you an overview of the Dup15 syndrome, not the clinical components of it, but more the genetics and the molecular pieces of it. So, and when I talk about the Dup15q syndrome, I mean IDIC15s and interstitials that include this region on the Q arm, which is the Q12, Q13 region, or Q12, Q13, as a lot of the parents tend to say. As a certified geneticist, I have to say Q12, Q13 because that's how I learned it. But there are other duplications that occur, and these, I stole this little image off the website, and they are in the flanking regions that are closer to that centromere part, which is that little constriction and beyond it. And they don't cause the same kind of symptoms. So, and they don't include that critical region. So I'm not going to be talking about those kinds of duplications, but they also do occur. And so just to clarify that the Q13 and the Q11 regions are not really part of this. So there's a lot to take home from this image. What you see here on the top, let's see, is an image of chromosome 15 sort of laid on its side. And this region that we're interested in here is blown up at this level. And that's all the little bands that we look at, the light and the dark parts. The next level down are, if you see those numbers, they are the megabase positions on the chromosome. And the reason I point those out is because when you've gotten your clinical reports, you probably saw on your array results that there was a duplication that occurred from maybe position 21,363,212 to some odd position. And that's telling you where on the chromosome those occurred, because megabases are millions of bases. And so that's 20 million bases, 21 million, 22 million, and so on down the chromosome. And then those are just giving us absolute positions on the chromosome. And below that, the breakpoints are marked there in color. And those are similar to the things that Jessica was talking about this morning, where, or maybe earlier this afternoon, I'm not sure exactly when her talk was, where she was talking about the repetitive regions that occur on the genome that mediate rearrangements between chromosomes and within chromosomes. And these are regions of DNA that are pretty big. You can see how long they are compared to the millions of bases up here. So this is what, almost 500,000 bases of DNA that are repetitive DNA that actually match a region down the chromosome. So here, this segment, which is highlighted in sort of this maroon color, matches this region down here on further down the chromosome. And so, and the blue and yellow down here. And then lastly, down at the below are the genes that are found in the region. And again, this yellow box is supposed to highlight the critical region. And again, I stole this off the Dup15 website. So if you want to go back and read about it later, you can go to their website and read some of their information. So one of the questions I always got from parents is, well, why did this happen? You know, what made this happen? So this is sort of a repeat of some of the stuff that you heard this morning, but essentially those repeats can set up chromosomes for rearrangements that are not normal. But what normally happens is that during the formation of an egg or a sperm is that chromosomes exchange material. And that's how you pass on, you know, information from both of your parents to your children. And so if the crossover, which is what we call it, occurs equally, then you end up with two new chromosomes that have exactly the same material. They haven't gained or lost anything, but now they have, you know, genes that came from one of your parents and then genes that came from your other parent. And so, but they haven't gained or lost anything. But what happens because of those repetitive regions is that sometimes there's a misalignment between one of those repeats and another one. And in this case, the formation of an interstitial duplication, this breakpoint 3 region aligned with the breakpoint 2 region. And when that cross occurs, it leads to duplication of this critical region in tandem. And so, and then the reciprocal product would be a deletion of it, but that actually causes a whole nother syndrome. So we won't go into that one. So when this gets passed on, it would be a duplication of that critical region. So there would be an extra copy. To explain why an eidetic forms requires me to show you a more complicated picture of what chromosomes actually look like during formation of eggs and sperm. And in this case, it's actually only in the egg, because eidetic 15s tend to only form during egg formation. And so when an egg is, and when a gamete, so an egg or a sperm is forming, the chromosomes have actually duplicated themselves. And so they look like those little X's that we show you when we show you pictures of other chromosomes, when you look at them clinically. And so that cross occurs between the arms, just like I was showing you when I just showed you a single arm. But instead of being straight down, it actually has stuck itself together. And when it does that, now it's got, the two chromosomes are actually hooked. And they've got to pull themselves apart to go into two new cells. So they start getting pulled apart, and now you end up with this new version of a chromosome that actually has two of those little centromeres, or those little constrictions that are important for sorting chromosomes out. Then as they start to end up in cells, into new eggs, the one egg will end up with one normal 15. The other egg will end up with a normal 15 plus this new chromosome that has two centromeres of the eidetic 15. And the reason that they come together and you don't just end up with an eidetic as a single, in a single cell or a single new egg, is because it may actually happen sometimes, but we don't see it because the product wouldn't be fertilizable. And so if it did, it wouldn't be a viable thing. So the only way that it would survive fertilization and make a baby would be if it landed with a full length chromosome 15. So with all of those different breakpoints, you'd think we'd see all kinds of permutations of what kinds of chromosomes are made. And indeed, we did see this. This is a slide from one of my graduate students, Nick Wang, who has now gone on to bigger and better things. And this is two of the forms of interstitials that we saw that tended to fall between breakpoint 1 and 3 and 2 and 3. And this looks like there were a lot more 1 to 3s, but in reality, this is 3 people from one family. So it's really pretty equally distributed. And that's an important point, is because this was a familial case up here. Then in the eidetics, we saw 2 very common forms, which were the breakpoint 3 and breakpoint 4/5s. And those are shown here. And what I wanted to make a point here is that this one is much smaller than this one and contains a whole lot less genetic material, because this one has, they both have 4 total copies of the regions through breakpoint 3, but this one extends now with 4 copies through here and a 3rd copy through here, which you can see through dosage here. This is 4 total copies, 3 copies, and this is 4 here. So, and we thought when we started our study that we were going to say, severe kids, mild kids. Wasn't that way. We saw actually milder symptoms in both groups, severe symptoms in both groups. So it wasn't that size was the indicator for clinical severity. The other thing we did see, though, was that we had 2 kids who had the misfortune of actually having 6 total copies because of pretty complicated rearrangements. And those kids were clinically, I mean, the best way to describe it is really devastated. And they had, so we know copy number does matter. We also have kind of gotten that feeling because interstitials tend to be milder than the kids with IDIC, and then this was clearly a very complex syndrome. The other thing we noticed was that we had 2 kids who had interstitial rearrangements that looked exactly like our IDIC cases on just array data. And so we wanted to make this, it's a reason that we absolutely need to be sure that people get chromosomes done. And I think, Jessica, you wanted to say something here?
I would just agree with the spectrum, and it's not always that the number of extra copies you have correlates to what we see clinically, which is something that actually came up in the other session. Someone had a question about that. So our clinical experience is the same. Similarly, a lot of what's out there is interstitial versus isodicentric, makes a clear difference in terms of the spectrum, but we also have seen a pretty broad spectrum with that as well. So we certainly have individuals who have that interstitial duplication who have similar clinical features as an isodicentric. And the only reason I wanted to bring that up is that a lot of times when people come to us, there's an assumption made about the testing. So it comes back with a duplication and there's an assumption made that it is from a parent, and based on clinical features, the provider makes the referral and says it's isodicentric. And just by looking at the child, we can guess. And if you see a lot of kids with duplication 15q, you can be occasionally right, but there are definitely exceptions. And so you do need to do the additional testing to determine the structure.
Exactly. We had one of our interstitials was sent to us as an isodicentric because they'd only done array data, and and it was important because that actually ended up being a familial one, and so then we were wanted to be sure that was followed up on, and so you definitely want to make sure that you do additional studies. So from there, the question is typically, so how are they diagnosed? Well, most of these kids have had an array or something. For a while, people were doing something called MLPA, which identifies the extra dosage. And those are like SNP arrays or the array CGH, which is what my lab was doing, and which is kind of another old format that people were using a few years back. And depending on when your child was diagnosed, you probably had one of those 2 things. And then that showed that there was actually extra copies that included the critical region. So then what needs to be done next? Well, we need to know what kind of duplication was it. Was it a IDIC or is it an interstitial? And then our other question is if it's interstitial in particular, or if it's a kind of complex rearrangement, where did it come from? And we use a test called the methylation study to look to see where it came from. And when I was doing clinical genetics, we always did parental chromosomes as well. it sort of happened somewhere along the way. And I am a geneticist, so I'm very clear on the fact that I think all kids who have this diagnosis should see a geneticist, but a lot of them were being diagnosed in neurology or in developmental peds. And as a researcher, sometimes I was the first geneticist that they were seeing. And it's, I think it's important that clinically all kids are being followed by a genetics team so that they can get the multidisciplinary components covered, because we tend to know more about the syndrome and keep in touch with the things that are going on with the syndrome. So what does array data look like, and what did the doctor see that showed them that you had an extra copy or that your child had an extra copy? So this is an array, again, from my lab, but it's essentially what a clinical array looks like. And it's just focused on the 15 here instead of across the whole chromosomes. But so on 15 here, this is showing dosage. Up higher, it goes up on the line, the dosage increases, and the little boxes are colored to show you how many copies there were. So that would be 4 copies, 3 copies, and this is 2 copies, which is what we would expect. And in this case, we saw essentially a signal that was consistent with it being 3 copies across the board up until we got to the position of breakpoint 3, and then it dropped back down. And this turns out to be one of our interstitials who had a breakpoint 1 to 3 duplication. And in this case, we saw it was the signals that were running in the break into the 4 copy range until we got to breakpoint 5, where we saw it drop back down to normal. So this looked like a breakpoint 5 duplication. And then to confirm that, we did what I was saying, what should be the next test, which would be chromosomes with fish. And so here's the chromosomes spread out. In fish is something called fluorescence in situ hybridization, which is what that stands for. And what you do is you take a little piece of DNA, which from this one, we took that piece of DNA that matched right there, and we fluorescently labeled it green, and we essentially, you make it stick to the chromosomes. And here, the red one marks 15 for us, so we can kind of cheat. And this is the 2 normal 15s. And the green shows us that on the IDIC, yes, indeed, we're seeing the breakpoint 5 signal there. So we knew it did extend that far. And then the last question is, where did it come from? And why do we want to know if it's maternally derived or paternally derived? Well, virtually all IDIC 15s are maternally derived. There have been 2 so far out of all of the years that these things have been studied that have been reported ever that have come from a paternal chromosome. And those have all been in the face of multiple kind of craziness things going on in the genome. So, and they didn't have symptoms that looked at all like IDIC 15 or Dup15 syndrome. So, and we don't know why that's happening, but there's been much debate. But for interstitial duplications, it's critical that we do the methylation studies. And the reason is that they can be maternal or paternal in origin. And if they're maternal in origin, they have the risks of the Dup15 syndrome, and it can be varied in severity. The other thing is that, as I pointed out when I had 3 kids from one family, is that they can be familial. And it can be, if they're paternally derived, so if they came on the paternal chromosome, it can be that that they have very mild symptoms, or they can be typically developing or have such mild symptoms that they didn't really know that they were, because the mild symptoms can be either some sort of autistic features, which you actually might, if you knew me very well, you might think I have some autistic features, and and sleep disorders, including parasomnias, which are things like sleepwalking and things like that. So those are more common in people with paternal duplications. So, but they don't stand out as being a huge syndrome. So if a mom has a paternal duplication, she won't necessarily know it, but if she passes it on, then her kids are at risk for having the Dup15 syndrome. The other thing is that if it's a paternal interstitial triplication, which is what we saw, that looks a lot like an IDIC, they can have symptoms that look just like the severe IDIC 15 cases. So it's another reason that you need to know what the parent came from, or where the chromosome came from. And then lastly, we had one patient who had a paternal translocation who had pretty interesting symptoms that sort of overlapped with Dup15. And so again, it's hard to predict what the outcome is going to be if you start rearranging within this region. So this is just to kind of give you a quick overview as to what a methylation study is. And again, I stole the image off the Dup15 Alliance website, so you can go back and do the homework again if you want to look at it. And what it does is it takes advantage of a gene called SNRPN on chromosome 15 that has a modification to the DNA that's called methylation. And the methylation on SNRPN actually causes the gene to be silenced or turned off. So on the maternal chromosome, which is the pink one, it is unmethylated and the gene is active. And on the paternal chromosome, it's methylated and it's silent. And if you, no, I'm sorry, I said that backwards. On the paternal chromosome, which is the pink one or the purple one, it's unmethylated and it's active. And on the maternal chromosome, it's methylated and it's silent. And on the paternal chromosome, if you have an interstitial duplication, you end up with an additional methylated copy. And if you have an IDIC, you end up with 2 additional methylated copies. So to take advantage of this, the test this is shown as a visual to kind of give you an idea as to what they look at. But essentially, they look at the ratio of the methylated signal to the unmethylated signal. And this is from 2 people with duplications, 2 people without duplications, and then this is a person who has a deletion, so that they only have the paternal copy. And the test is actually done using a computer-based way rather than done using this gel-based way, which is what we were doing. And it's much more accurate, so it can give you a ratio of the 3:1 versus 2:1 is more accurate, but it still was enough. This can tell you whether there looks like there's an increase in the maternal signal and can tell you whether or not it's maternal or paternal in origin. And so again, why does it matter? Well, molecularly it matters because we think that it's those maternal transcripts or maternal genes that are giving us the clinical outcome. We think that they're at least contributing to it. And some of the genes that are active only on the maternal chromosome are being highly studied in this disorder. And then the other major reason is because of recurrence risk in families. So we want to know where it came from and then to be sure that the parents don't have copies. And this is the other reason that I always did parental chromosomes, particularly if there's an interstitial rearrangement. And then that's sort of it for me. I showed you some data from Nick and from Dahai. Actually, I didn't mention that it was his data, thanks to the Dup15 and thanks to everybody who participated in my studies. There was like 100 of them and they did a wonderful job. I love them all. And then here's to Jessica.
So I just wanted to summarize with kind of the way we work up duplication 15q syndrome. So usually if someone presents with delays in development or neurodevelopmental concerns, we'll start with that chromosomal microarray, which looks for any missing or extra parts or pieces of chromosomes across all 23 pairs of chromosomes. If it comes back that there's an extra part of chromosome 15, then there's a little bit more work we need to do, both to determine structure and also to determine parent of origin. And so we recommend looking at the chromosomes and doing those FISH studies as a next step to understand structurally where are those extra pieces. And then finally, we can do DNA methylation to understand parent of origin. I also recommend often doing, as I kind of said a little while before, doing parental testing For recurrence risk, I think primarily it's helpful to do that. It also can provide some reassurance to families. But as we roll out with clinical trials that are coming for duplication 15q, which is super exciting, a lot of the companies doing the studies and pharma is going to want to know specifically what that change is. And so we're getting a lot of questions about this, and certainly I'm happy to talk with anyone on an individual level, but this is kind of the progression of how we fully understand this region of chromosome 15. So with that, we'll be happy to take any questions. Yeah, so I'm just going to repeat the question to make sure that Carolyn can also hear. So the question was, early on during diagnosis, no one had mentioned testing parents or concerns. And so what would be the utility of testing parents, if I understand correctly? Carolyn, do you want me to answer you?
Well, it depends in part on whether it's an isodicentric or an interstitial. And for, I mean, isodicentrics are almost all sporadic, but we've had, as I mentioned to when I was talking to you earlier, we've had a couple of cases where there was something else, a rearrangement that the parent had that predisposed to the formation of the IDIC. And so I typically liked to check parental chromosomes just to be thorough and make sure there was nothing that was going to increase the risk, because otherwise the recurrence risk for an IDIC is low but not zero. And to be completely clear that there was not anything else that was going to change that risk, I wanted to check parental chromosomes. And particularly if it's an IDIC, check mom's chromosome.
I think, yeah, particularly if you're thinking about having more children, and as I've talked to some of you in the audience already, it could even if you're not planning on having additional kids, it could have implications for your other children as well. If there's, for example, something balanced that's predisposing to having a slightly increased risk of having a child with duplication 15q. So I do it often for reassurance, and I tell families it's unlikely that it's going to be abnormal, but if it is, then at least we have that information to give a very more absolute answer as much as we can about recurrence risk.
Wait, Jackie, hold on.
For this, so Carolyn.
Oh, okay. If you're testing the parent, would you recommend just testing specifically for like my daughter's IDIC? So would you recommend just testing for IDIC, or would you recommend kind of a bigger microarray to test for other things?
I actually would recommend, and maybe Carolyn would have a different answer, but I would recommend actually the karyotype, where we look at the parent's chromosomes under the microscope and make sure that we don't see anything different or one of those translocations on that. Would you agree with that, Carolyn?
That's what I would do too, because I would just look for, I would just do a karyotype. You know, I thought of one other point I wanted to make, is that for any kid with an interstitial or an IDIC whose clinical symptoms fall way outside the realm of what we know for Dup15 syndrome. And I'm thinking of features like, I had, in the patient I'm thinking of had like abnormal fusion of his skull. He had cleft palate. He had abnormal fingers. They're things that we just don't see in this syndrome. Do additional testing. Having Dup15 does not preclude you from having something else.
Sorry.
And so
Sorry, go ahead.
it was just a matter of, we went for years not beating around saying this just doesn't look like, it was an interstitial case that just didn't look like it was way too many things that just didn't fit, but nothing else came up. And then finally, when the whole exome sequencing thing came out, he was able to get tested and we figured out what his other disorder was.
I would just add to that, that if you do determine that the duplication originates from the dad's copy or paternal copy, then if the clinical features are outside of what you would expect, which we can see some features like mild autism or delays in development, it is also worthwhile to just consider doing additional testing. Because we actually just had a case where the child's actually 14 and diagnosed duplication 15q, and we did a methylation test when I saw him in clinic, and it was paternal in origin. And then I did the broader testing, and he actually has a whole another syndrome called Smith-Magenis. So we see cases where it is worthwhile to pursue additional testing.
Any other questions?
Yes.
So just a quick question is I'm like, sometimes explaining her genetic condition to others, the question gets asked like, well, was it passed on from mom or dad? So how do you answer that? And it kind of goes along with that testing piece. Like, did we, for lack of a better term, did we give it to her? So, or what's the probability that one of us have it and maybe don't realize it and passed it on?
That's where parental testing comes in. But
Yeah.
Yeah,
So
sorry, go ahead.
can I answer that?
Sure.
Okay, so when we look at parent of origin testing, there's two different levels of that. There's one that says which chromosome is that? Is that the mother's chromosome or is it on the father's chromosome? And every baby got one copy of chromosome 15 from each parent. So she had to get 115 from you regardless. So if she has a maternal chromosome 15 duplication and you had to give her a 15. So it's not your fault. You had to give her a 15. And what I didn't point out on that slide when I meant to do it is those alignments that happen that cause the duplications, they occur very, very early in the formation of an egg. They actually occur so early that moms were in their own mother's uteruses when that lining up occurred. And so you can't, people will say, well, what did I do wrong? Well, you didn't do anything wrong. If anybody did something wrong, your mom did, because you were still a fetus when those chromosomes lined up. So and if you may not, if you have totally normal chromosomes and it's not something that you passed on, it's just it's on the maternal chromosome and it's not. And if they want to know whether or not did you give it to them, say no, it's an IDIC, it's a sporadic event.
If I might, I'd like to add a little to that. My name's Janine Cody. I run the Chromosome 18 Clinical Research Center, and I'm going to give a talk tomorrow. But actually, one of the things I always tell our families is that 50% of conceptions are chromosomally abnormal. And 90% of those don't implant. So you don't even know you're pregnant. So it's only a small portion that even move on to implantation. And then of those, 90% are miscarried. So, we're the lucky ones who have the survivors, okay? So it happens to everybody all the time. Nothing specific caused it. There's no data that shows there's an environmental insult or anything like that. It's just biology. So
I would
if-
just add to that, whenever you say genetic or you say syndrome, people assume that it was inherited from a parent. So it was a change that was definitely inherited from a parent who also has that change that they passed on in all of their cells. And that's a misconception. A lot of these changes happen in the individual sperm or the individual egg, as Carolyn said, very early on. So in that first phase that I mentioned briefly in the previous talk, meiosis 1, which is that early phase where you're going from 2 copies of every single chromosome in all of your body cells down to 1 copy in your germ cells or your eggs and your sperm. So these changes occur frequently. The way the genome is set up with all these duplicated regions, we know these changes occur frequently. But just because there's a genetic condition doesn't mean it was inherited from a parent. And I think that's something we need to continue to work on educating people about.
Any other questions? Well, thank you so much, Carolyn. And oh, got one? You're going to ask a question for what? Oh, okay.
You already have a son who is isodicentric. And then they also have a daughter who has autistic-like tendencies, who is older. Would you recommend testing mom and dad, sister, or all 3 of them?
Did you hear the question?
Did you
Do want
follow?
me to answer?
I can
Yeah.
say it again.
So
if
I
it
can
doesn't make sense.
repeat it
Okay.
too.
Are they
Yeah.
full siblings?
If needed, and I can also answer it. The question was, they have a child, as far as I understand, they have a child who has an isodicentric duplication 15q, and another child who has autism. Is that an indication for parents to get tested?
And also
And-
the sibling with the tendencies that has never been tested.
Gotcha. And also the sibling. So I can answer first, which is that I think if there's concerns, neurodevelopmental concerns about the other sibling, that sibling definitely warrants a genetic workup in case there's anything there is to learn that could help us better help that child. So I would recommend doing the typical workup that we do for neurodevelopmental concerns. It would be helpful to know a little bit more about what testing was already done in your child who has a diagnosis of isodicentric duplication. And I'm happy to talk more about. Yeah, so I'm happy to do that because sometimes that's helpful, but it depends what workup's already been done in the rest of the family, whether it's a possibility that that individual could have a duplication of 15q as well.
All right, I am going to close close questions because the next speaker is actually in the Netherlands right now. So it's like 2 o'clock in the morning and he's waiting to talk. So Dr. Dewis is going to hang around. So you're welcome to ask questions directly to her and show her your genetic report. But we're going to say a big thank you to Dr. Dewis and Dr. Shannon.